Hello, in this video we will be discussing DNA replication and what it entails. DNA is stored in the nucleus of a cell. DNA replication occurs during the S phase of a cell cycle when the cell is getting ready to divide. This is to ensure that both daughter cells contain the same exact genetic information after cell division. DNA is arranged in a double helix structure. So firstly, the topoisomerase enzyme unwinds the double helix to relieve tension, strain, and supercoiling of the structure during DNA replication. As shown, there is an unraveled section of DNA, and now the replication process is ready to begin. Note the DNA structure. DNA has a sugar phosphate backbone. The deoxyribose sugar is covalently bonded to the nitrogen bases. Covalent is strong and common. Phosphate is only bonded to deoxyribose. Weak hydrogen bonds are in between nitrogen base pairs. Cytosine and guanine only bond with each other. Likewise, adenine and thymine only bond with each other. That's the base pair rule. The helicase enzyme is the unzipper. It breaks the weak hydrogen bonds. Single-strand binding proteins keep the two strands separated. Here we see a replication fork shape. The two strands are anti-parallel. Leading strand runs in 3' to 5' direction. Lagging strand runs in 5' to 3'. The primase enzyme begins laying down fragments of RNA known as primers. The primers are laid at the starting points of synthesis, otherwise known as 3'. The primers are initiators. They let the polymerase enzyme know where and which nucleotides to place. The polymerase III enzyme begins continuously synthesizing the leading strand in a 3' to 5' direction. It adds nucleotides and brings in nitrogen base pairs based on the base pair rule. Adenine and guanine are double-ringed purines. Thymine and cytosine are single-ringed pyrimidines. The lagging strand synthesizes discontinuously. Polymerase III adds nitrogen base pairs in the 3' to 5' direction on the lagging strand, while the replication fork moves the opposite way. As polymerase III synthesizes the new strand and adds nitrogen bases, it has to go back to the start where DNA is starting to unravel again. So RNA has to add another primer so that the polymerase III knows where to start again. This causes Okazaki fragments. The replication fork unwinds fast. So the reason why there are Okazaki fragments on the lagging strand is the polymerase enzyme has to keep moving up to where the DNA keeps being unwinded quickly in order to build the new strand. New primers have to constantly be placed in order for polymerase to continue adding nucleotides, explaining the reason why these fragments are there and why they start with primers. DNA polymerase I replaces RNA primers with DNA nucleotides. DNA polymerase II comes in to proofread the nucleotides and possibly repair any incorrect base pairs. This helps prevent mutations. The ligase enzyme comes in to hold the Okazaki fragments together. It joins the fragments that start with primers together until the strand is completely new and complete. It acts as a glue. Here are the two identical DNA strands complete. As you can see, DNA has an anti-parallel nature. One strand runs in the 3' to 5' direction and the other strand runs in the 5' to 3' direction. This duplicated DNA has a semi-conservative nature. One of the two strands are newly synthesized, and the other strand belongs to the original DNA molecule. Now that the cell has duplicated its genetic information, it is ready to divide.